This morning, Dr. Nalini Mallikarjuna has been kind enough to give a seminar summing up her maybe 30, 35 years of experience in wide hybridization with examples of Pigeonpea, where we had one of the most successes. Dr. Nalini joined ICRISAT. I tried to know the date from you, didn't tell me. In 1980s, 70s. Okay, so maybe in late 70s, because when I joined, she was already here. Okay, so at that time she was working, it was called tissue culture, groundnut cytogenetics at that time, and to see the sight of a groundnut plant being grown from the somatic tissues was a beautiful sight, and that photo appeared on several of the ICRISAT's annual reports and other publications in the beginning, because that was the kind of high science at that point of time. And of course, that also laid the foundation for wide hybridization and genetic engineering at ICRISAT. To develop genetically modified plants, the first thing you have to know is the tissue culture. Once you have the tissue culture protocols, then you can go for transformation, gene insertion, expression, and of course, you want the final product, which has all the good traits in the world. So Nalini, after that groundnut work, continued with the work on groundnut, on chickpea and pigeonpea. She has many successes to her credit, including introgression or having a successful cross between the primary gene pool and the tertiary gene pools in pigeonpea, chickpea, and also in groundnut. So that's a unique achievement in the sense that once you want to make such successful crosses, you don't make one, two, five, or 10 crosses. You have to make 1,000, 2,000 crosses. And if you're lucky, you get one seed, two seed, and then further to be able to grow that seed into a plant, and that plant then again produce a fertile pollen or produce a seed, further seed to have further progress in introgression of those useful genes is a very difficult task. She has been successful in achieving many of these unique successes. A lot of that work has been published in the international journals nationally. I also had the benefit of sharing her experiences working with her. We worked quite hard to develop Helicoverpa resistant chickpeas and pigeonpea by drawing upon from the gene pool from the wild relatives, largely successful with the secondary and of course tertiary gene pool in pigeonpea and Platycarpus. So this has been a remarkable success. So let us all first congratulate Nalini for her singular achievement before I give floor to her. So it's all yours, please.
Thank you, Dr. Sharma. And it's a pleasure to be before y'all. As Dr. Sharma said, yes, this pigeon pea joined later into the group of white crosses. We started with groundnut. We have a long-standing experience in groundnut, but pigeon pea and chickpea came much later. Thank you very much for the lovely introduction. I'm grateful to you. And I offered to give this seminar, and ICRISAT was kind enough to say, oh yes, please go ahead. And so today I'm going to talk to you. Although LSU, I gave this as Conquering the Gene Pool, they wanted to put a little milder title, but I like putting this title because it shows my enthusiasm. It almost meant like, conquering. And I'll tell you why as I take you across with it today. First of all, I must thank my collaborators at ICRISAT, my friends, my colleagues, who have played a major role in all the success stories that I'm going to tell you today. One way or the other, they are important, and I thought I must start with thank you and then go ahead. Why should we really go and conquer the gene pools? It's a difficult thing. As Dr. Sharma said, you do all those crosses, sometimes you end up getting sterile plants, but why should you do? Sometimes you have got to do. Why? Because if you take an example of pigeon pea, everybody says any paper you read, it has a narrow genetic base. Why? I'm just giving you an introduction because Pigeonpea evolved from a single species, which is Cajanus cajanifolius. Or probably some of you who are doing molecular biology will say, no, Scaraboides. But either way, it was one of the species that really evolved and gave rise to the diploid cultivated pigeonpea. And the second bottleneck, it is an Indian origin. Had it originated in Australia, India, Myanmar, and Africa, probably it would have had a little broader genetic base. Single origin. And of course, any crop, there is farmer selection. Farmers selected. They are the first plant breeders. And of course, you have the really very important plant breeders at ICRISAT and everywhere, who did fantastic work, got the pigeonpea to the state it is today. But then all these processes narrows the genetic base because you select for traits and you get rid of something that you don't want. And this leads to the narrow genetic base. And superimposed on this is the climate change, land degradation, energy crisis, water scarcity. This all leads to why we should go and look into the gene pools. And I might tell you now, one of the best ways to bring in variation into plant species is by wide hybridization. Some of you may question why. There are other methods. But here, you transfer traits en masse. I'm not saying it's a lovely story, but who says life is easy? You get a lot of unwanted traits, but there are methods now, today, where you can get rid of what you don't want, take that en masse, and take what you want. And you get to have a lot of traits at your disposal you can work with. Coming to the really gene pools of pigeonpea, and we are following, normally we all of us follow the method of Harlan and Dewitt, 1972, but that works fantastic for us. The primary gene pool species are closely related. This is all based on the relationship, crossability with cultivated pigeonpea. That's how the different species are placed in different gene pools. And if you look at it, the Quaternary gene pool has only related genera. And I'll take you through all the gene pools. And that's why I have written Conquering the Gene Pools of Pigeonpea. Recently, we had a success, and that really excited me. I said, before I leave ICRISAT, I must share this with the group. Primary gene pool, a very important one. There are so many excisions. Anything which you call pigeonpea, anything you call Cajanus cajan, anywhere in the world, it's in primary gene pool. Breeders have done fantastic work. You have so many varieties. Pigeonpea would not have been as it is today had it not been for those work. But I'm not going to read into detail. It's good enough if you know this much. If you go to the secondary gene pool, there are various species in the secondary gene pool, and I showed it to you. Whenever we start an experiment with the wild species in a gene pool, it's not like we just started with these two. It was a broad-based experiment. We worked with Scaraboides, we worked with lineators, we worked with acutifolius. Lanceolatus is a very latest entrant, and I'll tell you later about it. But we narrowed down to acutifolius because whenever we start an experiment, we have a dialogue with all my collaborators in ICRISAT. They're also learned, so and cache their knowledge. And they said, take something which has not been used before, try to tap those traits which we want. So we took this wild relative from the secondary gene pool. It's native to Australia, and people had not used it in a big way. And we knew it had a report of disease and pest-resistant traits. And who did all this? Many of my colleagues at ICRISAT screened it for various traits. And I must tell you something about it. Although it's a secondary gene pool species, not all it doesn't work like you take and you just cross and live happily ever after. If you reverse the cross, it doesn't work. So when I say secondary gene pool species, you have to take it with a pinch of salt, work it yourself, and see what happens. Relationship shows that it's close by. And when we started the experiment, with the permission of my entomologist colleague, they said I could have just put it as broaden the genetic base. In those days, it was very important that you worked for and you showed your progress. So we said Helicoverpa armigera, which is a very important. Pod borer is a big problem in pigeonpea and chickpea. They said, okay, you work for pigeonpea because the cultivated pigeonpea doesn't have resistance in a big way. Try to transfer it. So we picked up this acutifolius. And what happens? Why Helicoverpa and why pigeonpea? You can see that nice fellow sitting inside and gobbling the pigeonpea pod. It's there. And what happens to the pigeonpea? You have all exit holes. Pigeonpea is useless. Imagine what happens on the farmer's field. The crop is useless, no yield. How does this happen? Happy Helicoverpa, sad piece. And this is, of course, I didn't do it all by my own. As I said, it's a collaborative work. You work with people. get results. Okay, we have had publications on this various, and I'll also take you. And pod borer resistant Cajanus acutifolius, we crossed it, and it's not like overnight we got all the species. I mean, this is painstaking work of a few years. We crossed it. We are very sure now these lines, at least on ICRISAT center, many of these lines. Now I would urge you to look at only the blue-violet line. I'll talk about the other one later. So we have lines which have the resistance, and not only pod borer resistance. I said white crosses to introduce variability. We also transferred high seed weight. Cultivated pigeon pea, the seed weight is something like 8 to 10 grams, 100 seed weight. But we had here, to our surprise, 18 to 20 grams. And if I would put the seeds on the table, they would just roll out like the pearls. I was tempted, if I had the opportunity, to name them as Hyderabad Mutyalu, because Hyderabad is famous for pearls. And we got this lovely pigeon pea. Even if someday we do, we should give that name. And we have at present 70 such advanced generation lines which look like cultivated pigeon pea. They are stable. We have also given ICPL numbers to it. And we also, what we did at one stage when we knew they had pod borer resistance, we went and looked if it had fusarium and SMD. And my collaborators, as I showed you in the photograph, they played a major role. And as you can see, I would just ask you, you don't have to bother to read the whole table, but just look at these lines. It had fusarium resistance, SMD, ICRISAT isolate resistance, PB. We don't have Phytophthora blight here, but that's the fun of working with people, and they seem to have some amount of resistance to Phytophthora blight. And they also have large seed weight, high seed weight. And look at the value that we get when you use the wild species. You have en masse transferred all of this together. As I said, there are 70 such lines. They're available in ICRISAT. Please use it. Okay, the spillover here was, as we were working, you always look at, you go with people to the field. We noticed, the male sterility in this material. And for the first time, we identified the A5 CMS system. And this has been reported. And the nice thing about the system then was, it was the only system available on cultivated pigeonpea cytoplasm. When you have the wild species in pigeonpea, if you read the story, there are many reviews. Most of the system is on wild species cytoplasm. This was the first time, and the breeders told me, go ahead and use it. You have got a fantastic system. We need it in the heterosis breeding program. Give it to us. Work with it. We did it. We identified the maintenance and restorers, and we have handed it over. Many times, till you got the maintenance and restorers, we were thinking, it was also a learning process for us. We did the whole thing, and we have given it to the breeders now. And as I said, this is a late entrant. I won't talk too much about it because it's my PhD scholar's work. Sandhya Shrikant, who is in the audience. See, we had this experience with us. The first time we took a little longer. This is not about 3 years old. There was this lanceolatus we picked up. We crossed it. I must just say, we are going to report the A9 CMS system. She has to write, I have to read, and this goes into publication. And A9C was recently discovered. The beauty of the system is, again, the system is on cultivated pigeonpea cytoplasm. I said, sky is the limit. I tell people who come to ask me for my opinion, work with different species. They have a repertoire of genes. Please use them. I mean, if you have the time, you don't know what you will discover. I mean, this is what it is. We have identified some sort of maintainers and restorers, and we are going to, once Sandhya does her work, this will be handed over to the breeders. Because we have finished what we have to pre-breed with it, and we don't want to meddle anymore with it. Now, coming to the tertiary gene pool species. Most of the wild species in the tertiary gene pool, I think today I'm just going to be a bit bold, live apart my modesty, and tell you, ICRISAT is the only institute who have worked with the tertiary gene pool species, succeeded in crossing, got the derivatives. What did we achieve? We reported the A7 CMS system. They have plasmoglamous flowers. I'll tell you what it means. The lines have pod borer resistant, pod fly bruchid, all field resistance, Phytophthora blight resistance. I might just tell you that Cajanus platycarpus is one of the only wild species which has P3 race resistant to Phytophthora blight. And volubilis, I'm not going to talk too much about it because it's my PhD scholar's work. She has been lucky. It was also nice to have her because With the experience, we were able to achieve a lot with the work. I'm sure her thesis will be very interesting. And I'll take you through. This is how it looks, Phytophthora blight. Try to just remember this one because I'll come to it. What you're seeing is Helicoverpa. It's trying to, nibble on the pod. It's not successful. It's not growing. Like I showed you the first phase where there was a fat, healthy Helicoverpa. So this is what happens with the wild species. And I think my esteemed entomologist colleague will tell you this is antibiosis mechanism of resistance, which you can transfer. And when you work with the tertiary gene pool species, there is a lot of challenges to face. And with it, and as I said, we were one of those who developed this system, harmonized pollinations, embryo rescue techniques, tissue cultures to go with it. As a result, we have, but I just tell you a little bit, F1 embryo rescue. That itself is bad. It takes so much of your time. When we had the BC1, I said, no, not again. But we had to do the embryo rescue again. Because we had such an important material at our disposal, we just didn't want to give up. Don't give up. That's another thing when you work with the white crosses. It's very challenging. What I'm telling you is all the success stories, but there's a lot of hard work that goes into it. But I think it pays when you work hard. And we had the embryo rescue in BC1. Then we had mature seeds. We back-crossed 4 times. We had advanced generation progeny. Beautiful. We were excited because they looked like Pigeonpea, but they were shorter. We had already experienced having one of them with acutifolius, which is medium duration. This was short duration. And we had this field trial. And one of my colleagues challenged me and said, what guarantee you have your Platycarpus genome sitting there anymore? You're back-crossed 4 times. For all I know, it would have gone away. I said, but look, you have these traits. He said, maybe it's because of the variation. We took it as a challenge. And I must thank AGL, the present AGL, but in its past form, they did the DART analysis. And what I would ask you is don't bother to look at the whole thing, but just look at these pink lines. There were about 1,000 markers. I've just taken a snapshot to show you. This is the Platycarpus genome. And after 4 back crosses, it just followed the pattern. Whatever amount of DNA had to be there were there. And when you do a white cross, you will have some recombination. And even those DNA stretches were there. And this was exciting. And it was very satisfying. I went back to my colleague and said, well, look, you challenged me, but that was fantastic because it made me work more. And now it proved that, yes, we still have the Platycarpus genome. And that has contributed to these traits. Derivatives show variation for plant growth. They're small, beautiful. One of the lines is a dwarf, but it is susceptible to Phytophthora blight. We weeded it off. When you start with the base is almost like this pyramid. You have to eliminate all your material. It's not very easy to throw away, but you can't keep all because there's no way how we can store. There was a point when my whole lab was with seed packets, and I would dread if anybody would visit my lab because they said, why is all the seed packets lying here? I never had the heart to throw. I said, I don't know when I have to use it. But very reluctantly, recently, we had to clean up, and we did all that. But we have retained only the best material that we should have, which you can take forward. Please, anybody wants to use it, please use it. And we have, as I said, different Platycarpus. We had beet seed color seeds and also Helicoverpa resistance. Now, please look at this column. This is the Helicoverpa resistance. Sorry, it should have been 0 to 10, but we didn't look for 0 resistance. When I was talking to people, the experts in Pigeonpea, they said, don't look for 0 resistance, look for low. We want is low damage. Because if you have 0 for all the traits, maybe you won't have any yield at all. I took their advice, did go for zeros, but many of them have very, very low resistance. We also had a A7 CMS system in Platycarpus. This was first reported by us. And this system has both pre-meiotic and post-meiotic pollen abortion. I'll tell you what it means. That's why I'm saying it's a very interesting system. And this we have handed over to Pigeonpea breeders. What I mean by this 2 meiotic systems abortion. Pre-meiotic, this is where the pollen mother cells are formed. And there is a meiotic division, which is a reduction division. Then that's very important. If the reduction division doesn't take place, exponential raise of chromosome numbers would have been there. We would never look like what we are. In all living systems, that happens. Most of the living systems, higher living systems, it happens. Gametes are formed. There is abortion at this stage, and there's a cut. It stops. Pollen mother cells don't develop further. In this system, we also found, if at all there were escapes, there was post-meiotic abortion. Tetras formed, but they all stuck together. They didn't form the gametes. They didn't form pollen grains. So they didn't give rise to any pollination. So that was a lovely CMS system. Not just this. The system also had clasmogamous flowers. All of you must have heard clistogamous when my Pigeonpea, colleagues have given the seminar. Closed flower. It helps to breed, Pigeonpea variety because Pigeonpea has a small window of cross-pollination. But this has clasmogamous flowers, not only pre and post meiotic anther abortion, but it also had non-dehiscent anthers. What do I mean by this? See, I want to show you. This doesn't look like Pigeonpea flower. This looks like a rubiaceous flower. If you know a bit of taxonomy, you'll know how rubiaceous flower looks like. And these flowers were such, and you can see this is how the pollen grains were. And what did I mean by, there was no dehiscence of anthers. If at all any of the pollen grains were there should be the line of dehiscence here, which opens up and releases the pollen grains for pollination or whatever. In these, those region thickens up. There's no way how pollen grains can be released. We looked in, why is this system so peculiar? And we're excited to find this. The nice thing about is, I know we don't in ICRISAT work on storage pests. Bruchids are very, very important. All of you who keep legumes at home, especially chickpea and pigeonpea, and you forget and keep it in a box, you get those insects inside and makes the seeds useless. So we said, and one of my visits in China, I looked at, they were working for Vishya and for bruchids. I came back and said, when I'm doing interspecific hybridization, when the whole en masse traits we transfer, why not test this for bruchids? And we found, some of the wild species that we tested had better resistance than the cultivated. What I did, I also had the interspecific derivatives. We screened some of them. Some of these are Platycarpus lines, some of these are Scaraboides lines, and this is acutifolius line. We found there was better bruchid resistance than the cultivated, and even if the eggs sat on the seeds, they did not hatch. It did not help them too. So some sort of antibiosis mechanism of resistance. We have published this, but there's a lot of scope to work on this constraint. Platycarpus, there are 140 Platycarpus, advanced generation, pigeonpea-like looking lines with us. They've all been given ICPL numbers. They've all been pre-bred, ready to use. And it's in ICRISAT, it's both in breeding as well as in cell biology unit. Any of you are interested, please take it and use it. We also screened this material for Fusarium and SMD. We found some of the lines had Fusarium resistant, SMD resistant, and Phytophthora blight low damage. I must tell you, all these 140 lines were selected based on their resistance to Helicoverpa. That was the rider. If it doesn't have Helicoverpa resistance, they don't stay in the cell biology lab. So we screened it, and I've just taken a snapshot. There are various lines which have SMD resistance, as you can see here. We have few with Fusarium wilt, but we put with all the three constraints. I might just change gear. I know it's Sandhya's work, but since I'm giving a review sort of a work on it, this is a very recent entrant. When we started work long, this is about 3 years old work, 2.5 to 3. She should correct me if I'm wrong. Volubilis, it was only reported for a CMS, A3 CMS system. They used it as a female. They got the progeny, they were sterile. It was not continued further. We used Volubilis. It is very, very challenging. You can see how the hybrids are. Some of the seeds were germinating even in the seed. That's why I said, when you do with the white crosses, you have to be all eyes and sometimes ears. And you can see this is the hybrid. This is the cultivated, okay. And this is the wild species. And once you look at it, once you work with it, your eyes tell you. And that I say it's your experience. It's just like how you recognize your own people. We knew it was a hybrid, but it was very challenging. Okay, we did the SSR analysis, and we knew it was a hybrid. This is the wild species, this is the hybrid, and this is cultivated. Okay. And we got the F2, and when I had this material, this is the F2 material. This was a dwarf. I said, where did he come from? I said, we didn't want a dwarf. I had to think over when I had the material. So what I do when I have this is I go to the experts. I went to the breeders, Dr. Samir Kumar in the audience, and I said, look, we have this short one. And when he looked at the material, he said, it's very exciting. Just go ahead with it. And before we talked to him, we had also ratooned it once. We said, no, I think something is wrong. Let's ratoon it and see how it grows. Outside, it was a dwarf. Inside the glasshouse, it was a dwarf. It gave rise to seeds. We got the F3, all dwarfs, F4, dwarfs, but with more branches. Then he said, this is something very important. And we have consistently got dwarfs. We don't have to do anything with it. So I told Sandhya, once you finish your work, hand it over the breeders and let them make use of it as early dwarf, and they have a little more seeds, high yielding ones. Coming back to the gene pools of Pigeonpea, primary I introduced, secondary I said what progress we made, tertiary I just now talked about it. This is the quaternary gene pool. Actually, this was a challenge. Sometimes when you talk to people, like-minded people, sometimes, it's very interesting because they said, you cross a quaternary gene pool species, you have achieved. I mean, probably this was a passing statement, but I took it seriously because we had the material for some reason. I said, why not we cross and see what happens? We crossed, we succeeded. And that's the beauty of it. As I showed you, there are various genera in the quaternary gene pool, Rincosia, Flamingia, Paracalyx. I mean, there are so many. I have to go back to the table. But we picked up Rincosia because Rincosia is very interesting. The tribals grow it. And some of the species have very important traits. Anti-cancer properties, anti-inflammatory properties. I said, okay, that's good. Let's see if we succeed. We did. And crossing, hybrids were obtained. It was very challenging. You look at the center one. That told me we had a hybrid. But that was not just enough because it was very, very interesting and challenging. We also did the SSR analysis. We knew what we had. At this point, we had a meeting. Then Dr. Gowda said, no, this is very, very interesting. I just wanted to send it to, a small publication because that was the time I said, only a few months left for me to leave ICRISAT. He said, no, this is not just yours. It's an institute achievement. Go ahead and work further. We went and did molecular cytogenetics, genomic in situ hybridization, and this shows all the green ones are Rincosia. I think it's a FITC labeling. All the red ones, I think it's Tropedium iodide. And this is metaphase, this is anaphase. We knew we had it. And that was exciting. But the story doesn't end here. You have to work further on it, but we are reporting this one. In summary, species from the secondary, tertiary, quaternary gene pool have been crossed successfully with Pigeonpea. What did we do? We diversified the cytoplasmic base. We contributed 3 cytoplasms, A5 from acutifolius, A7 from platycarpus, more recently A9 from lanceolatus. Disease and pest resistant stable lines. Some of the lines I didn't show because I'm sure my young colleague would work further. We did a small waterlogging experiment with an apprentice, and some of them withstood waterlogging. What we did in the glasshouse, they developed lenticels. We said, what is this? This has waterlogging too. And I must tell you, platycarpus also has salinity. We did a very tiny experiment, and I'm sure the traits are there. Please, people should work on it or use it. And I didn't really tell it, but I just wanted to tell you so that you know what it has. We have also had successfully tertiary gene pool species. It's not a fluke because we have crossed 2 of them. We have stable hybrids with us, which people can use it in the improvement program. Dwarf and early plant type, high seed weight and number. I must thank the cell biology staff because sometimes enthusiasm runs wild. It's okay with me because, maybe I'm passionate about it, but, sometimes you tend to push your people, So this is an occasion for me to thank you all. I don't know if you are there. Deepak Jadhav, who was a scientific officer with me, a fantastic gentleman, and all the others too, and the students, of course. I don't know, maybe I'm just lucky. I just get the right people. They're as enthusiastic as I am. And if not for that, we would not have achieved so much as we have. Anyhow, thank you very much for your passion. In summary, Okay, stable pre-breeding lines. I've told you all this. Okay, I pressed the wrong one. This is the end of Pigeonpea story. I have very important story to tell you about grandiose groundnut, groundbreaking research in groundnut. Would you like to hear? No, I know what you mean. Thank you very much for your patience, and I would be glad to answer any questions if you have. Please do. I'm told by Dr. Gowda that Dr. Shivali Sharma is going to take up from me. So this material in the cell biology will be with her. Yeah. Also the chickpea and the groundnut material. All this, some of them which are advanced lines in Pigeonpea, they are with groundnut breeding because I didn't realize then, but we have to use it in the TL2 program, from accessing genes from the wild species. So they have it. Hopefully, probably, they will multiply it. That's a request. So between the two of them, they should have. And cell biology, there's a lot of material. Please, anybody who wants, take it from there. I have also been telling people, anybody who came to me and anywhere I gave a seminar, I said, this material is available even for the groundnut, especially the synthetic. Please use, because if you don't use, what's the fun? We have taken so much pains to develop it. That's what I do. We did that. That's the reason, with a level of confidence, I said. When we looked into it, they're cultivated Pigeonpea type pots. I've not done, we did a small experiment in those days to take the, cross section and look at for the trichomes. So it didn't have that, one of those trichomes, I forget. The trichome work, yeah. So, but yeah, no, they just look like Pigeonpea. In fact, they're very soft. That's one of the things we look. But you can experiment for, according to me, it's antibiosis. Because as I showed you in Platycarpus, what you said may be right, could happen to Platycarpus. But if you look at the material, they just look like cultivated Pigeonpea. Yeah. Field resistance and first of all, cross them. You have lineatus lines, which has high protein. And I tried to see if any of these had high protein. Not too much. There's not a big difference. That's why I didn't put high protein lines. Not terrible difference. It's not like 28, 30, like compared to Pigeonpea, which is something like 20, 19 to 21. At the most, what I've seen is 23 or 24. So I didn't have the heart to say you have high protein lines. At least I have not. Also, sometimes there's a limit how much you can do. But in the advanced lines, I've recently got it analyzed. And I'm at this juncture, I'm in the process of writing manuscripts for all of them, for Platycarpus, for Cajanifolius. Hopefully, I was just planning to do before I leave, but even maybe before I leave the campus, maybe I would take, it becomes too much, in the end, because so many things you have to do administrative, otherwise you just can't sit and write papers. But the data is there, that's what I want to do. And it also helps the other people who are going to use this material. It should be ready-made, That's the most, information is everything. If you don't have it, how will anybody use it? So that's the thing what I want to do. Put it in papers, publish it, and hand over the material. And that's the reason why I said the seminar should be there. I don't know if I answered your question, but that's what it is. We should look at it. Now that we saw it, see, this was a small bit of an experiment when there was an apprentice. I saw the bruchids somewhere. As I said, I saw this experiment in China. We just did a trial and we published a small paper in the journal. It's a publication. That's why I put it here. Please take it because, and bruchid is not one of the constraints that we have put in our, I don't know, whatever document. We have not put it anywhere. In fact, I tried at one point. Oh, I've already been doing enough. So they said, after this. So, but if there is an opportunity, please, much can be done. Yeah, once you have the material, sky is the limit what you can do. I mean, within, intra, you should have an aim why you're doing it. You could cross within secondary gene pool, but you still stay in the wild species domain. I didn't even bother to do it. I was very clear. My goal was to get the traits into Pigeonpea. If I did cross in the secondary gene pool, I would still remain there, unless it was difficult to put it into the cultivated Pigeonpea. Then probably I would take secondary, cross it to tertiary, tertiary with quaternary. But I didn't have this issue in Pigeonpea. I didn't even want to digress really. Probably you can do it if you want to make a mapping population, go round about it. You could do it, but my aim was get Helicoverpa resistance. That was assigned to me, and it was very clear. We used to get questioned. Okay, you've done fantastic work in white crosses, but what use of it is to us? We have been asked this question. So I kept Portboro, as my whatever, and worked for it. But if you have any other intention to cross within, probably you could. I didn't bother to do it. Can I add up to that? There are two things. I knew your chickpea. I knew why you were asking, but this was Pigeonpea, so I didn't go. There are two ways. One is you could do that. The second thing is there are techniques in place. We have developed it at ICRISAT. Other people have used it. Australia and Canada have used it. Embryo rescue tissue culture. We get the hybrids, but they're very fragile. They don't withstand the transfer to soil. This is all published. If somebody can work on it, fantastic. And I've also written in my note, which I'm supposed to hand over, chickpea needs a lot of input. Do it. Reticulatum mechanosperma only two wild species which we have crossed. I did develop all this, but I knew, last 2 years I knew I wouldn't have so much time to do on that and money. You need to have what your growth chambers, so expensive. I said, no, I give up. But if somebody has all the resources at hand, and I think you all have, now that you have so much money in the project, probably you should do that. Go both ways. I don't know when you succeed. It's always good to have a multi-directional approach.
Many of these wild species, in both tertiary and secondary, these gene pools have accessions with more than 30% protein.
30%?
Seed protein content. That's a thing to explain. In this presentation, I have seen some labeling errors for ICPL, ICPW, and ICP. Please correct them because they are having some identity problems.
Thank you very much. I always get sometimes ICPW. Yeah, that used to be the original one. If I've taken the seed, I keep on sticking to it, but I get confused. I agree with you. I'll try to see that.